Hello, good afternoon, everyone. My name is Pedro Raposo, and I'm a curator at UniProt. In this presentation, myself and Antonio Locke, another curator at UniProt, we are going to talk about the tools integrated into UniProt's website, such as BLAST and Align. We are going to discuss how you can access these tools in order to get the most out of them and how to explore its results. To start with, UniProt is the world's leading high-quality and freely accessible resource of protein sequence and functional information. It has a reliable data, as we have teams of expert curators reviewing literature in order to describe what a protein does. UniProt is a massive resource, and it currently has 250 million proteins that cover more than 1 million different taxa. We provide a plethora of information ranging from protein function, catalytic activity, gene ontology terms, pathways, subcell location, etc. And we are going to provide sequences alongside stable identifiers as well. So besides protein information, we also have integrated some tools in order to ease your analysis when you are in our website as well. UniProt is a consortium that is composed of 3 different teams, and these are EMBL-EBI from the UK, the Protein Information Resource from Washington, DC in the US, and the Swiss Institute of Bioinformatics from Switzerland. And together we coordinate our efforts to provide you many services from our website, as well as vast knowledge on protein information. So just as an introduction to the different UniProt resources, we start by importing information from external genomic resources, such as DNA or GenBank, for example. Then the information provided goes to UniPark, our archive resource for protein sequences. Then we transform those sequences into protein accessions in the UniProt knowledge base, or UniProt KB. We group proteins by their organism of origin, creating proteomes. And finally, we provide data sets for cluster proteins based on sequence similarity in UniRef. But focusing on the UniProt knowledge base, this is composed of 2 different data sets, SwissProt and Tremble. So SwissProt contains records that are reviewed, which means that the curator has read literature with experimental data on this protein, and it has added that information to the entry. We can identify these entries by the golden icon with the star. And at the moment, SwissProt has over 500,000 records. And the other database is Tremble, which contains unreviewed proteins, but these are annotated through automatic annotation systems. And at the moment, we have 250 million records. So sequence comparison have been used by biologists for a very long time, and they're essential to address many questions. They are used for characterization of novel protein sequences, not only by inferring protein information, protein function by homology, but also to identify conserved regions that contribute to that function. And these alignment tools are also used for characterization of protein families and to explore evolutionary relationships between species. They are also used for curation of genome annotation to address many different biological questions. So 2 of the main tools are the BLAST and the Align. On one hand, BLAST finds local regions of similarity between sequences, and on the other, we have the sequence alignment, which compares sequences to find regions of similarity. So with BLAST, you'll get information to understand what the function of the protein is and if it belongs to a specific protein family. With the sequence alignment, you will get information to understand if specific regions or motifs have a particular function. and it will allow you to visualize them inside the alignment. So now that we know what these tools are used for, we are going to see them, how you can access them through the UniProt website. So on the left page, you have our homepage, and if you zoom in at the top of the page, you'll have the tools displayed there. Another way to access these tools is if you scroll down a bit on the page, you can see these 4 different tools here. You have Blast Align, you have List Map IDs, and the Search Peptides. And you can access them through that. You can also access the tools inside the protein pages. So one way of going into a protein entry is to search them by the search box you can see here. So once you open our homepage, you are welcomed by this search field in the middle of the page. And here, this is a free text field, which means that you can add any word or set of words and search. And you'll get a list of proteins with those terms inside that page. However, you can be a bit more specific with the search that you are doing. So if you click on Advanced, this panel will open, which allows you to be a bit more specific on the criteria you want to search for. So for example, if we want to search for proteins with a specific EC number, then we can go on the first field, write EC, and from this list you can select function enzyme classification EC. So when you click on it, then you can add the EC number you want to add. And the same thing can be done with the other fields as well. So for example, here we are going to search all trypsins in Homo sapiens. So if you click search, you will be welcomed by this results page with 25 entries. So there are 25 human proteins with this EC number, and each row here is one specific protein. And if you click on it, then you'll have access to the protein page. So in the protein page, you have access to the tools that we mentioned previously. Not only that, but it is in this page where you can find all the available information on UniProt that UniProt can provide to this protein. If you look at the left panel, you have the different types of characterization of this protein. There is a section on the function, a section on the different names and details about the taxonomy in which this protein was found in, a section in the subcell location where the protein exists, information on post-transcriptional modifications, expression, interaction with other molecules, its 3D structure, family and domains of this protein, the sequence itself and its isoforms, and also similar proteins to this one. So from here, you can navigate where you are interested to. So if you go to the sequence section, you can use the canonical or isoform sequences as queries to blast and align as well. And further down the page, you can select if you want to blast certain domains only of the protein. You can also select proteins you want to analyze from different protein lists, and you can also select proteins throughout the website and add them to a basket. So once you have that basket selected, you can open it and you can see the proteins that are in there. And you can select those to blast and align as well. So you have different forms of accessing this tool and selecting the different types of proteins. So now that we know how to select, I will start to cover first BLAST, which stands for the basic local alignment search tool. And as the name says, it finds similarity between sequences. And this lets you infer the function, do phylogeny studies, and identify members of the same gene family. So once you access BLAST, you'll get this page where you can add the protein sequence itself on as the header, but also on the second box, you can add the protein sequence. And afterwards, you can select the target database in which you want to blast against. You can add some taxonomic restrictions saying that you are only going to blast against human, for example. And you can add a name to your blast job. So here you can see the different databases you can choose to blast against. You have UniProt knowledge base, which contains SwissProt and Tremble. Then you can select, for example, if you want to blast against reference proteins plus SwissProt. And next to it, you have the restriction by taxonomy. And below that, you also have some advanced parameters if you want to look into something a bit more specific. And after that information sequence, you can start the run blast. So after that, you will be redirected to a dashboard where all the current running and complete jobs are displayed. And once the job is finished, you can click on it to see the results. So this is the results page. We limited the results to 250 alignments with this blast, and these are the details displayed here. So on the right side of the page, we have the alignments themselves, where if you click on it, it will show a more detailed mapping. And on the left side of the page, you can choose how to filter the results, being this by the alignment score, identity, or eValue. You can filter for reviewed and for unreviewed proteins, filter by taxonomy, by length, and other characteristics as well. So on the top bar, you can select different tabs to get more information on the taxonomy distribution and the distribution of the score, identity, and eValues as well. Also, you can use the results outputted here as inputs for a new blast or a new alignment. Now let's talk a bit about the align tool. So this is used to align two or more sequences to find conserved regions of proteins and to infer homology and evolutionary relationships. And it uses it uses classical omega as its basis. So similarly to BLAST, once you have access to the align tool, you get this page where you can add the UniProt identifiers to the first field or the sequences themselves on the second one. And you have some advanced parameters here as well. And you also can hit run align. And again, you are going to be redirected to this dashboard in which you are going to wait for the results to be processed. And this is the align result page. So I usually, when I come here, I click on the overview option here to be able to see the total alignment. And if you do that, you can get the clear picture on the whole alignment, and you can adjust the zoomed window to get a larger or smaller alignment for the individual amino acids on the bottom part of the page. So from here, you can investigate the sequences to understand what regions of these proteins are actually conserved. So for example, if you are interested on serine and threonine, you can highlight them by clicking the highlight properties and select serine threonine. So you can see here that this has been highlighted now in the alignment, so it's better for you to visualize. Another example is if you want to have a look into the active site. So you can click select annotation and type active site, and then click on the first protein, the protein that you are interested in. And here you can see in the alignment that an active site was found on the 431st position. And if you scroll there to see the alignment, You can see that this marker this marks a cysteine in all three sequences, which means that it is conserved in all these proteins. After analyzing your alignment, you can also download it in different formats. You can open these in multiple alignment software such as JALView or Mega, for example. And on the other tabs above, you have access to a phylogenetic tree, a percent identity matrix as well, a text output format of the alignment where the sequences are shown alongside a line of symbols that tells you how the alignment performed on each amino acid position. The next step shows you the parameters used in this alignment and also the job ID in case you want to revisit this page or share with your colleagues. And in the last tab, you can find how to run this exact alignment using curl if you want to run this job through a command line. So let's go through an example to have a better grasp on these tools. Draculin is a protein that was isolated from the vampire bat that acts as an anticoagulant of the blood coagulation cascade. So let's consider we want to know if this exists in other mammals. And if so, what are the function of those homologs? And let's see if they also have 3D structures associated with them. First, let's use the UniProt search bar and look for the term Draculin. This is the result page where you can see that the first hit corresponds to the protein coming from the vampire bat, and you can see this on the last column where it says organism. So if you click on it, then you have access to the protein page, and from here you can click blast. So this will open this page with the sequence in the second field already, so you can run blast. And this is the result page we found where you find multiple hits. So in order to better digest these outputs, let's narrow down our results by using some filters. So let's select only the reviewed proteins to confirm that the data we are looking for has been curated. So on the left panel, you have a section says status reviewed 15, which means that from this list you have 15 different proteins that have been reviewed and are in SwissProt. And after clicking on that button, you are going to select these 15 SwissProt, and you can tell that all of these come from mammals, including humans. We also can answer our second question on what the function of these homologs are. And here we can see that all of them are lactotransferins. Also, note that you can see some details on each alignment at the end of each alignment. So here you'll find three values. The first one is the identity percentage of the alignment. The second one is score. And the third one is the E-value. Now let's see if we have 3D structures associated with them. We can select the filter 3D structure, and we can see that 8 out of 15 have these 3D structures. And once you open the human protein that was listed there, you can read the function comments in more detail. And in this case, this is an iron binding transport protein. You can also see and interact with the 3D model structure as well. And because the human lactotransferrin is an iron binding transport protein, A following question we can ask is if our Draculin protein also has iron. We can verify this by seeing if the iron binding sites are conserved between these two proteins. So let's go back to our BLAST result and select an alignment between Draculin and human lactotransferrin. Click on the alignment itself. And after this, you go to the bottom of the page to select the annotation, and you can select binding site. So here you can see some amino acids from the human being highlighted as binding sites. You can also verify that the same amino acid is present in Draculin, our query protein, in the same position. So if you click in one of these, you can see that these are in fact binding sites to iron, which means that yes, our Draculin is also an iron binding protein. So just to highlight two additional useful tools in our website, this is the peptide search, and with it you can find all UniProt proteins that contain a specific peptide. This is useful when retrieving information on mass spectrometry studies, and this tool gets only exact matches, and you can find up to 100 peptides independently on each search. So on the top of the page of our website, you have access to this tool as well. And on the first field, you can see that we've selected two different peptides, and we are going to run the peptide search on these two sequences. You can add the peptides on the first field and restrict the taxonomy on the second field. And as usual, you will be redirected into this dashboard, where once the job is complete, you can click on it to be redirected to the results page. So finally, the ID mapping tool is a tool that allows you to get proteins by using a set of UniProt identifiers or other identifiers as well. So this is a very useful tool. So you can paste your collection of IDs in this first case, in this first field. So in this example, we are going to add here on, we are going to select a list of PDB entries, and we want to search them in the UniProt knowledge base. So here you can see that we have three PDB identifiers, and we want to see which identifiers we can see in the UniProt knowledge base. Then we can run this tool, and once it's completed, it will provide you the different IDs, and you can directly access them to see the protein pages. And the same thing can be done by trying to convert UniProt knowledge base IDs to ensemble IDs, for example. So here you can select the different databases. We select from UniProt KB to ensemble, and at the end you will get the correct ensemble IDs as well. And so with this, we had an overview on where to start searching proteins, what types of information does UniProt provide to this, and how to navigate the results and protein pages. And finally, how to use BLAST Align and some other tools as well in order to expand your analysis. So if you have any questions or want to learn a bit more about UniProt, you can visit the Help Center by clicking Help in many pages, in many different places scattered around our website. And also you can directly send us an email and we will assist you promptly. And with this, I would like to thank the UniProt team and our funders.
Okay, so we have a couple of questions that Antonia has answered. So hopefully everyone's happy with those answers, but also a couple ready to be asked now. So let me have a look at them. Right, so the first question that we have is, would you be able to elaborate a bit more on the BLAST algorithm itself? So, for example, just a bit on how BLAST works. I understand that you can't sort of take us through a demo step by step at the moment because, as you mentioned at the start, but if you can provide any more information on that.
Yes, so BLAST is going to BLAST is going to start with two sequences, the target and the query sequence. The query is the one that you are looking for, and the target are one of many proteins that exist in our database. And with it, it's going to start to look into the query sequence and compare to the target. And it's doing this by using a matrix of substitution. So this matrix is going to essentially tell you how similar different amino acids are biochemically. So you have different matrices that serve different purposes. So there are some matrices that you can use if you want to search for homologs that are very distant, or you have matrices that you can use when you want to be a bit more specific and have a bit more sensitivity on the alignment. So once you have chosen that matrix, then the first amino acid is going to compare, the first amino acid of the query is going to be compared with the first amino acid of the target. And if it has a good score, then it will move on to the second amino acid of the query, and so on and so forth. So if you have gaps between an alignment between the target and the query, then there's going to be a penalty in the score. And if the score goes too low, then the BLAST, the alignment is going to stop. So this will determine how much coverage you are going to get between these two proteins. So the better the score, and this is the score that you can see in the results in our website. So you have mainly three outputs for each alignment. You have the score, the coverage, and the value. The score is the one that I just mentioned. So the score is given by the substitution matrix in every single amino acid, and it's the sum of all of this. The better the score, the better the alignment is. So this is a very brief summary on how BLAST works.
Great, thank you. We also have a question that asks a bit about how BLAST can be used. So let's jump to that one. So could you use BLAST to infer the evolutionary order of mutations? Can one determine conserved regions and determine what mutations occur when for a specific protein family?
I guess it's possible, but you need to be aware of what type of mutations you want to observe. Because if it's a SNP, maybe it will not be, BLAST will not be able to detect all the very, very small mutations. But if it's something large enough, then yes.
Great, thank you. So we have another question that asks, if I want to download a specific sequence within an alignment, for example, from 90 to 120, which represents a specific domain, is it possible to do that?
So it's possible for us to align domains and families from Interpro, but I don't, and Antonio, correct me if I'm wrong, but I don't think we are able to BLAST a section of the protein without manually actually removing the amino acids when it comes to the BLAST itself. I don't think we have that. So in that case, if you want to search a specific region of the protein, you are able to download the FASTA on the protein, and you can select the region, and then you can BLAST against our website. I would recommend that.
Great, thank you. Having a look at the Q&A at the moment. I can see someone just asked about the recording, and yes, we will email out with the link to the recording tomorrow, so everyone who registers will get that link. Okay. And then I think the last question we have is, if I want to identify a protein's active site or binding site, is it possible to obtain this information from UniProt?
Yes, we do have that information. So inside each protein accession, you have a section on that page where you can see that information. So the information that I just shown in the alignment, where you can see if there's a binding site there or not, that comes from the protein page itself. So that information is there. You can have access to that as well.
Wonderful, thank you. So that looks like all the questions that we have for today.
I added an example to the to a protein that has an active site. In the, I don't know how those answers are shown to participants, but it's in an answer if people want to see how it's shown in the website.
I've just popped that into the chat as well, so everyone should be able to see that there.
And in the advanced search, it's also possible to search on active sites.
Wonderful, thank you. Okay, so I think if that is all the questions for today, we can leave it there. So I'd just like to say thank you very much to Pedro and Antonia for joining us for today's webinar, and also to all of you for attending and for asking your questions. Thank you.